This meeting is being recorded.
All right, welcome everyone. I am just super thrilled to see you out there in the little Hollywood squares of Zoom land, and thank you so much to Andy for taking on this this topic. It's garnered a lot of interest, and so hopefully folks will make their way. This is a Met How at Home program, and we saw that other villages were watching these TED Talk series, and so we did a few last year, and Andy has revived it with this topic today. So I will be handing it over to you, Andy. I'll let you finish your introduction.
Are you seeing the screen here?
Yes, I'm seeing your slide. The natural world we see around us is incredible, is what I see.
Okay, great. I can't see it, so it helps me to know. I'm just going to tune it in here a little bit more. Still sort of learning this system. It's amazing, Zoom. I guess that's one good thing that came out of COVID, right?
I know, that is one fun thing.
Okay,
Yeah.
well, good morning. I appreciate all of you coming. I'm pleased to have a chance to talk to you. My name's Andy Miller. I've been coming to the Valley since the early 80s, first as a visitor and a sometime resident. I have a place off of Cum Road and Winthrop, which I've owned since 1991. And, I think all of us in the Valley really love the natural world around us. That's what brings us here, as well as the community of people, Methow at Home. I've been a member for about 3 years. But today I'd like to take a little peek under the hood of the micro world of life that we don't see, which I just find really fascinating and beautiful as well. I originally sort of understood the interrelationship of many organisms doing embryology and looking at the embryos. Are people able to hear me? My system doesn't always work.
Yeah, that's great when you stay sort of in the middle and up close. Right. That's perfect.
So we're going to look at the elegant simplicity of the genetic code, which basically writes the symphony of the cascade of biochemistry of all life. And I used to be in biochemistry, and this book over here to the right, which was written by Walter Isaacson, sort of brought me back to studying biochemistry again. It highlights one of two Nobel Prize laureates. 1920 chemistry prize went to Emmanuel Charpentier from France and Jennifer Doudna, who's from the US and from California. And they developed a tool called CRISPR-9 genetic scissors. And it's basically a tool from the immune system of bacteria. They're both microbiologists, and with it, we can change the DNA of any animal, plant, or microorganism. And already, since it was really developed in the early, about 2013, 14, it's created a big impact in the life sciences for cancer therapy and promises to allow us to begin helping people who have inherited diseases. It's also, interestingly, when COVID came out, these two researchers mobilized an international group to create the two COVID vaccines, the Pfizer and Moderna vaccines. Normally, a vaccine takes about 4 to 5 years to develop in the traditional method, but in about 6 to 9 months, they were able to create a vaccine, basically using a system like the immune system of bacteria to create the vaccines, as well as the rapid COVID testing kits we've used, or most of us have used. But before we jump in, and we're going to watch a TED Talk given in 2014 by Doudna, Jennifer, I'd like to just go through some basic biology and microbiology so we can sort of understand more what this is about. All living things, other than small viruses which don't have cells, are made up of one of two types of cells. They're either prokaryotic cells, which is mainly bacteria, And for the most part, these are unicellular organisms or eukaryotic cells. These are much larger. Prokaryote means before the nut, and that's because these cells don't have a nucleus or a nut inside the shell. Eukaryote cells are the true nut. They have a nucleus or a nut inside. And we'll look at the morphology of these two different kinds of cells in just a minute. But prokaryotes are unicellular. They have no nucleus. Again, bacteria is the most common type of prokaryote. They have one long loop of DNA floating in the fluid of the cell. And records suggest that they're about 3.5 billion years old. They were sort of the first life that came to be. And they are felt to be the precursors of eukaryotes. Eukaryotes, on the other hand, are multicellular organisms. They have a nucleus. The DNA is in multiple strands. They're called chromosomes in the nucleus. And they've existed about 1.5 billion years. And as I said, there's good evidence. It was first just theorized, but now there's good evidence that eukaryotes ingested each other and then kept what they ingested inside, which became what are called organelles, and formed a complex cell known as eukaryotes. And this is a diagram of the two. This is the prokaryote cell. This purple is just a big long strand of DNA floating in the fluid, which is called cytoplasm. And that's the genetic material, deoxyribonucleic acid, for this organism that allows it to reproduce and run its metabolism. And they can be somewhat complicated, sometimes flagella, allowing them to swim. They have sort of a thick capsule. And that's in contrast to a eukaryote cell, which has this nucleus, which, early morphologists thought this was like a nut, a shell with a nut. Here's a shell without a nut, so prokaryote, eukaryote. And within this nucleus are these multiple strands, the chromosomes, DNA, that form that cell, as well as the larger organism, since there are multiple cells that make up most eukaryote animals. Inside the cytoplasm, the fluid, these other organelles occur, which again are thought to be old prokaryotes that were ingested. In this case, we have the mitochondria, which is the battery, which all the energy is stored in a eukaryote cell. Also interesting, chloroplasts are also organelles that are in plants. And as Dana Versali, one of the other Valley residents, has taught us, it's the chloroplast and photosynthesis, which is really the origin of all life, which again was an organelle that started in a eukaryote and created our atmosphere, turning it to somewhat oxygen instead of the toxic materials it used to be. So this is the place that we found this immune system that the CRISPR is made from. And again, because bacteria have lived for 3.5 billion years, they've been fighting viruses for a long time. And they created a very interesting system to fight viruses, recognize them, and then attack them and kill them before they kill the cell. And that's the CRISPR technology, which we'll again go into a little bit more. So something else that's important as we look at all this is the Human Genome Project. That was a collaborative international effort that looked at the DNA, mainly of the human genome, and wrote the entire genome or mapped it so that we could study it and learn about genetic diseases and also potentially help people. It was carried out not just in the States, but all over the world from 1990 to 2003. And it also looked at other organisms like tuberculosis, E. coli, important pathogens, hoping that we might be able to fight those pathogens by knowing more about its genetic origin. So what are the genomes? What is DNA? I look at it almost like a program in a computer. DNA is a file cabinet. It's the hard drive of life. And it's a language really of just 4 different codons or 4 different nucleic acids. The DNA, as we all have heard, is a double helix, and the helix is held together by these nucleic acids. There are 4 of them: adenine, which always pairs with thymine; cytosine, which pairs with guanine. And they create this lattice that holds the 2 parts of the helix together. And they are essentially a code by the order that they're in. And the triplets of these codes, which
I'll take it in the other room as soon as I'm done making my coffee.
are called codons. And these codons then basically are a language that prescribe for an amino acid to build proteins. And we'll talk a little bit more about proteins. But proteins are basically what form all cells and organisms, and they're written for by these triplets from the DNA. And they run the metabolism of the cells. Metabolism is basically the chemical processes that occur within a living organisms in order for it to maintain its life. And it's pretty amazing, from these 4 codons, or these 4 nucleic acids and the codons they create, that the whole language of the diversity of all carbon-based life is written. And again, as I say, the DNA is not very active. It basically is just a storage place for all this information. And the RAM for the, just a second, the RAM for cells, which is the random access memory in a computer, that's the short-term memory that goes around and does RNA.
Andy, just so you know, okay, you went off, your
Okay.
voice went offline for just a second there, so
Better
you
now?
might have
Better?
to repeat.
Thank you.
Yeah,
So RNA is the cousin of DNA, and it's a single helix instead of a double. And this shows us what those nucleotides are. Here's cytosine, which pairs with thymine, guanine with adenine, and you can see how they create this rib of the double helix. RNA has 3 of the same and 1 different, uracil, which then binds with cytosine. And you can see how these 2 can pair, and basically the RNA reads the data off of the DNA when it wants to build something. And so again, I sort of think of that as the RAM. It's the workhorse of the cell. It basically starts, goes in, a bit of the DNA uncoils, the RNA copies it, then goes out of the nucleus and goes into the cytoplasm and builds proteins. And this is another just diagram which shows the 9 steps, which go from the DNA, the file cabinet, to create proteins. So what happens is the DNA unravels, exposes its code. Transcription RNA, there are different kinds of RNA, comes in and copies that code. And then it goes outside of the nucleus, and that's called transcription because it's not changing the language. It's the same language. It transcribes DNA to RNA. It goes outside the nucleus to a ribosome, which is also a form of RNA. And then it's translated to another language, into the language of proteins. And that transfer RNA brings in, with the codons that we talked about, specific amino acids, which are the building blocks of proteins. So here we go. DNA transcribes to RNA, which is then translated to another language of protein, a different chemical, macromolecule chemical. And here we see the amino acids. Here's proline, serine, different amino acids. So what is a protein? Proteins are very complex structures, and now we've been able to look at them carefully and see how beautiful they really are. We've used 3 different techniques. We use nuclear magnetic resonance spectroscopy, cryoelectric microscopy, and X-ray crystallography to look at these. And basically what each of those are, we take a protein, we freeze it, and then we look at it very carefully, almost like a CT scan using X-ray or MRI to look at the entire structure. And proteins have three sort of structures that can be talked about. First is the primary structure, which is just the chain of amino acids. But those chains, the way that they interact, create ribbons and complex shapes, and then they fold to create a tertiary structure. And here to the right is a structure which is hemoglobin. Hemoglobin is in our red blood cells and carries oxygen from our lungs. It then combines with iron.
If you could move to the center again, Andy, sorry.
There are 4 pieces of iron in the this big complex. And basically proteins, there are about 8 different types of proteins that are built that run the cells. One are called antibodies, which we know are our immune system. There are also contractile proteins like myosin, which is our muscle. Enzymes are basically proteins that push different chemical reactions to occur. Hormones, such as insulin and glucagon, which we've all heard about, are proteins. Transport, which we just talked about, are proteins. So they basically, it's a cascade. I mean, it's interesting. We sort of know how DNA writes for these things, but we really don't understand what controls it all. Like, so you have all this code, it transfers, what really tells it how to build humans, because or any organism. So we really are we don't know a lot really about how the system works. We're learning how to control it a little bit. This next slide just shows different diagrams. This is hemoglobin. This is looking at hemoglobin. We can see there are 4 different units that individually form, then combine with iron to be hemoglobin carrying oxygen to the muscles, which have a similar type of structure called myoglobin, which also have iron in them. This is another look at it. This is with X-ray crystallography, the look. And then just to give you an idea of when things go wrong, this is hemoglobin, normal hemoglobin, the way it looks with the iron within it. This is hemoglobin in a person who has sickle cell anemia, and you can see how disorganized it is. This can carry two oxygen molecules. This can barely carry one. You can see there's only one iron in this sickle cell. And sickle cell anemia is one of the diseases that has been cured so far with the CRISPR technology. So we've been talking about CRISPR. This is our last slide before we get to listen to Jennifer. What does CRISPR stand for? CRISPR stands for C for clustered, regularly interspaced, short palindromic repeats. And during the Human Genome Project, these were found, and we didn't know what they were for a long time. In 1986, a Japanese student in Osaka was working to do look at the bacteria E. coli. You know, E. coli is what we see in our stool and often is a pathogen causing disease. So they were getting the genetic code of it to try to help fight it. And when he was looking at, he found these sequences of code, 29 long, that were identical, scattered throughout the genome. So that's the regularly interspaced. And they were palindromes. And a palindrome is something that's written the same forward and backwards. The example is the name Ada, which is spelled the same backwards and forward, or the term do ye see God. So D-O-G, do, God, ye see God. It's the same backwards and forward. And in these bacterial cells, there were these palindromes and then DNA coding in between them. They had no idea what that was. Later in the 90s, a Spanish PhD student was looking at Archaea, another single cell organism, and found similar palindromes. And finally, in the early 2000s, in the Netherlands, they were looking at the genetic code for tuberculosis, and they again found palindromes. And he's the one who made the term for it that you could remember. And it's from that, you can see that, it's never one person who does everything. It's a long process of learning and then understanding in how we make things go forward. So now we're going to go on and we'll take a look at this TED Talk, which I hope this introduction helps us understand a little bit, by Jennifer Doudre. This was done in 2014. In it, she's going to talk about the positive and some of the scary things that this CRISPR technology is allowing us to do. So I hope this will go. We'll see.
Your dedicated Cintas service reps help make sure your team has the essential products they need.
Is it the sound coming
Yep.
through?
Looking great,
Yep.
feeling confident, and ready
Sounds
to be
good.
their best. Get Cintas and get ready for the workday.
A few years ago, with my colleague Emmanuel Charpentier, I invented a new technology for editing genomes. It's called CRISPR-Cas9. The CRISPR technology allows scientists to make changes to the DNA in cells that could allow us to cure genetic disease. You might be interested to know that the CRISPR technology came about through a basic research project that was aimed at discovering how bacteria fight viral infections. Bacteria have to deal with viruses in their environment, and we can think about a viral infection like a ticking time bomb. A bacterium has only a few minutes to defuse the bomb before it gets destroyed. So many bacteria have in their cells an adaptive immune system called CRISPR that allows them to detect viral DNA and destroy it. Part of the CRISPR system is a protein called Cas9 that's able to seek out and cut and eventually degrade viral DNA in a specific way. And it was through our research to understand the activity of this protein Cas9 that we realized that we could harness its function as a genetic engineering technology, a way for scientists to delete or insert specific bits of DNA into cells with incredible precision that would offer opportunities to do things that really haven't been possible in the past. The CRISPR technology has already been used to change the DNA in the cells of mice and monkeys, other organisms as well. Chinese scientists showed recently that they could even use the CRISPR technology to change genes in human embryos. And scientists in Philadelphia showed they could use CRISPR to remove the DNA of an integrated HIV virus from infected human cells. The opportunity to do this kind of genome editing also raises various ethical issues that we have to consider, because this technology can be employed not only in adult cells, but also in the embryos of organisms, including our own species. And so together with my colleagues, I've called for a global conversation about the technology that I co-invented so that we can consider all of the ethical and societal implications of a technology like this. What I want to do now is I want to tell you what the CRISPR technology is, what it can do, where we are today, and why I think we need to take a prudent path forward in the way that we employ this technology. When viruses infect a cell, they inject their DNA. And in a bacterium, the CRISPR system allows that DNA to be plucked out of the virus and inserted in little bits into the chromosome, the DNA of the bacterium. And these integrated bits of viral DNA get inserted at a site called CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. A big mouthful. You can see why we use the acronym CRISPR. It's a mechanism that allows cells to record over time the viruses that they have been exposed to. And importantly, those bits of DNA are passed on to the cell's progeny. So cells are protected from viruses not only in one generation, but over many generations of cells. This allows the cells to keep a record of infection. And as my colleague Blake Wiedenheft likes to say, the CRISPR locus is effectively a genetic vaccination card in cells. Once those bits of DNA have been inserted into the bacterial chromosome, the cell then makes a little copy of a molecule called RNA, which is orange in this picture, that is an exact replicate of the viral DNA. RNA is a chemical cousin of DNA, and it allows interaction with DNA molecules that have a matching sequence. So those little bits of RNA from the CRISPR locus they bind to protein called Cas9, which is white in the picture, and form a complex that functions like a sentinel in the cell. It searches through all of the DNA in the cell to find sites that match the sequences in the bound RNAs. And when those sites are found, as you can see here, the blue molecule is DNA. This complex associates with that DNA and allows the Cas9 cleaver to cut up the viral DNA. Makes a very precise break. So we can think of the Cas9 RNA sentinel complex like a pair of scissors that can cut DNA. It makes a double-stranded break in the DNA helix. And importantly, this complex is programmable. So it can be programmed to recognize particular DNA sequences and make a break in the DNA at that site. As I'm going to tell you now, we recognized that that activity could be harnessed for genome engineering to allow cells to make a very precise change to the DNA at the site where this break was introduced. That's sort of analogous to the way we use a word processing program to fix a typo in a document. The reason we envisioned using the CRISPR system for genome engineering is because cells have the ability to detect broken DNA and repair it. So when a plant or animal cell detects a double-stranded break in its DNA, it can fix that break either by pasting together the ends of the broken DNA with a little tiny change in the sequence at that position, or it can repair the break by integrating a new piece of DNA at the site of the cut. So if we have a way to introduce double-stranded breaks into DNA at precise places, we can trigger cells to repair those breaks by either the disruption or incorporation of new genetic information. So if we were able to program the CRISPR technology to make a break in DNA at the position at or near a mutation causing cystic fibrosis, for example, we could trigger cells to repair that mutation. Genome engineering is actually not new. It's been in development since the 1970s. We've had technologies for sequencing DNA, for copying DNA, and even for manipulating DNA. And these technologies were very promising, but the problem was that they were either inefficient or they were difficult enough to use that most scientists had not adopted them for use in their own laboratories, or certainly for many clinical applications. So the opportunity to take a technology like CRISPR and utilize it has appeal because of its relative simplicity. We can think of older genome engineering technologies as similar to having to rewire your computer each time you want to run a new piece of software. Whereas the CRISPR technology is like software for the genome. We can program it easily using these little bits of RNA. So once a double-stranded break is made in DNA, we can induce repair and thereby potentially achieve astounding things, like being able to correct mutations that cause sickle cell anemia or cause Huntington's disease. I actually think that the first applications of the CRISPR technology are going to happen in the blood, where it's relatively easier to deliver this tool into cells compared to solid tissues. Right now, a lot of the work that's going on applies to animal models of human disease, such as mice. The technology is being used to make very precise changes that allow us to study the way that these changes in the cell's DNA affect either a tissue or, in this case, an entire organism. Now, in this example, the CRISPR technology was used to disrupt a gene by making a tiny change in the DNA in a gene that is responsible for the black coat color of these mice. Imagine that these white mice differ from their pigmented litter mates by just a tiny change at one gene in the entire genome. And they're otherwise completely normal. And when we sequence the DNA from these animals, we find that the change in the DNA has occurred at exactly the place where we induced it using the CRISPR technology. Additional experiments are going on in other animals that are useful for creating models for human disease, such as monkeys. And here we find that we can use these systems to test the application of this technology in particular tissues, for example, figuring out how to deliver the CRISPR tool into cells. We also want to understand better how to control the way that DNA is repaired after it's cut, and also to figure out how to control and limit any kind of off-target or unintended effects of using the technology. I think that we will see clinical application of this technology, certainly in adults, within the next 10 years. I think that it's likely that we will see clinical trials and possibly even approved therapies within that time, which is a very exciting thing to think about. And because of the excitement around this technology, there's a lot of interest in startup companies that have been founded to commercialize the CRISPR technology, and lots of venture capitalists that have been investing in these companies. But we have to also consider that the CRISPR technology can be used for things like enhancement. Imagine that we could try to engineer humans that have enhanced properties, such as stronger bones or less susceptibility to cardiovascular disease, or even to have properties that we would consider maybe to be desirable, like a different eye color or to be taller, things like that. Designer humans, if you will. Right now, the genetic information to understand what types of genes would give rise to these traits are mostly not known. But it's important to know that the CRISPR technology gives us a tool to make such changes once that knowledge becomes available. This raises a number of ethical questions that we have to carefully consider. And this is why I and my colleagues have called for a global pause in any clinical application of the CRISPR technology in human embryos to give us time to really consider all of the various implications of doing so. And actually, there's an important precedent for such a pause from the 1970s, when scientists got together to call for a moratorium on the use of molecular cloning until the safety of that technology could be tested carefully and validated. So genome-engineered humans are not with us yet, but this is no longer science fiction. Genome-engineered animals and plants are happening right now. And this puts in front of all of us a huge responsibility to consider carefully both the unintended consequences as well as the intended impacts of a scientific breakthrough. Thank you.
Jennifer, this is a technology with huge consequences. You pointed out your attitude about asking, you can call it a pause or a moratorium or a quarantine, is incredibly responsible. There are, of course, the therapeutic results of this, but then there are the non-therapeutic ones, and they seem to be the ones gaining traction, particularly in the media. This is one of the latest issues of The Economist. Editing humanity is all about genetic enhancement, it's not about therapeutics, right? What kind of reactions did you get back in March from your colleagues in the science world when you asked or when you suggested that we should actually pause this for a moment and think about it?
My colleagues were actually, I think, delighted to have the opportunity to discuss this openly. It's interesting that as I talk to people, my scientific colleagues as well as others, there's a wide variety of viewpoints about this. So clearly it's a topic that needs careful consideration and discussion.
So there's a big meeting happening in December that
Correct.
You and your colleagues are calling together with the Academy of Science and others. What is your expectation? What do you hope that will come out of the meeting practically?
Well, I hope that we can air the views of many different individuals and stakeholders who want to think about how to use this technology responsibly. It may not be possible to come up with a consensus point of view, but I think we should at least understand what all the issues are as we go forward.
Now, colleagues of yours, like George Church, for example, at Harvard, they say, yeah, ethical issues basically are just a question of safety. We test and test and test again in animals and in labs, and then once we feel it's safe enough, we move on to humans. And so that's kind of the other school of thought, is we should actually use this opportunity and really go for it. Is there a possible split happening in the science community about this? I mean, are we going to see some people holding back because they have ethical concerns and some others just going for it because some countries under-regulate or don't regulate at all?
Well, I think with any new technology, especially something like this, there are going to be a variety of viewpoints. And I think that's perfectly understandable. I think that in the end, this technology will be used for human genome engineering. But I think that to do that without careful consideration and discussion of the risks and the potential complications would not be responsible.
Now, there are a lot of technologies in other fields of science that are developing exponentially, pretty much like yours. I'm thinking about artificial intelligence, about autonomous robots, and so. No one seems, aside from autonomous warfare robots, nobody seems to have launched a similar discussion in those fields and calling for a moratorium. You think that your discussion may serve as a blueprint for other fields?
Well, I think it's hard for scientists to get out of the laboratory. Speaking for myself, it's a little bit uncomfortable to do that. But I do think that being involved in the genesis of this really puts me and my colleagues in a position of responsibility. And I would say that I certainly hope that other technologies will be considered in the same way, just as we would want to consider something that could have implications in other fields besides biology.
Jennifer, thanks for coming to TED. Thank you very much.
Thank you.
All right, are people still here?
Yep, I think we're all still here.
All right, well, here's, I guess, a little discussion afterwards. I'll come off after this, but there is a 3-day summit in London in March of 2023, again, where they're going to discuss this. In 2018, a US trained scientist working in Beijing shocked the world when he altered the genetics of 3 young girls that were just embryos. And that is what you can potentially do, is you can make designer humans. He was actually jailed by the Chinese government for doing this for 3 years for his unethical practices. So that's one of the real quandaries that we're going to be having to face with this and AI. Anyway, I would stop sharing here and maybe open it up for discussion.
Yeah, so go ahead. You can either use your hand raising function, or you could just come off of mute and jump in, or use the chat. So don't be shy. What are some of your thoughts?
Hey, Dana.
If you're speaking and we aren't hearing you, you're probably not off your mute. So
let's see. I have my hand up. But I don't know
sorry, I don't know. I guess I'm. Yeah, go Tuck. Thank you.
So I would just remark that this is not the first time humanity has faced difficult ethical issues like this with emerging technology. I think nuclear weapons in my field would be an example where many of the early participants became gravely concerned about the consequences of the emerging technology. And, you know, I think you kind of had, and I would say probably and effectively over the ensuing 80 years, have not not been successful in managing the ethical discussion. But I think you need to see in an awesome technology like CRISPR, you need to look at that through the lens of human behavior. And so there's the question of, is everybody going to be responsible? So if you knew what responsible behavior was, is everybody going to be responsible? I think the answer is almost certainly no. Human history just, is flying in the face of that. And so I think pausing for a discussion is an important thing to try to do, but I think you need to see the forces that are at work. And certainly in the case of nuclear weapons, the forces that were at work basically carried the day. And there are going to be forces at work, not only governments, but others who see advantage in CRISPR, who are likely to carry the day here.
I mean, interesting, Putin heard about this and immediately said, I'm going to build an army of super warriors out of it. So it does have a lot of potential for problems. And if we look at the scandals we've had recently with people getting their kids into schools illegally, if you have the ability to create your children, create advantages with your children, there's already one gene that they've identified in all sprinters, no matter what country sprinters come from in the Olympics, they have the same gene. There's another gene that creates very strong people. That human genome has really allowed us to better understand how we're put together and potentially make it possible to engineer people.
I mean, I think large, major intellectual advancements always are double-edged swords. And I would say humanity doesn't have a very good record in managing those double-edged swords. But you could pick steel or any number of major advancements in human knowledge and understanding, and they can be used for ill, and frequently are.
Mm-hmm. Yeah, thanks for that, Tuck. Dana, I see you ready to jump in here too.
Yeah, not necessarily, just emotionally reacting. It's a powerful subject. One thing that does come to my mind, it's been on my mind for a week or two. There's a line in a story by Joseph Campbell. It's something supposedly Buddha said. We don't know if he said it, and the line is, life is an ever-burning fire. Now that can be interpreted in many ways, but for me at this week, it's just out of control. Life cannot be controlled, and there's no way that we have the wisdom to control this particular technology, much less the other technologies. I mean, editing genes is not our actual problem. I mean, identifying what our problem is would be a conversation at best. But the vitality of the planet is diminishing. Species are disappearing. Ecosystems are disappearing. One could go on. So it just can't be. But in terms of life being an ever-burning fire, I don't see how you can help this happening. If it can be invented, humanity is so brilliant scientifically that humanity will invent it. But that doesn't mean we don't have the wisdom to use these things, and that's just the way it is. There's nothing that can be done about it. Anybody remember? I can't really capture it, but we have the insight of the gods and the emotions of the Neanderthals is a paraphrase.
Why are you blaming Neanderthals?
I don't
Maybe
know.
they were smarter than we are.
Neanderthals might have been. I think maybe they had more emotional intelligence.
It's all I can think of at the moment.
We wiped them out,
Well, by some measure, they were a far more successful evolutionary experiment than modern humans, right? Neanderthals were around for 250,000 years or something. You know, we've got maybe 60,000 in the game. You know, if you think humans are so brilliant, you know, who invented CRISPR really? Bacteria.
Yeah, yeah.
Right. So, you know, I don't think you necessarily want to count modern humans as a successful genetic experiment. I think it's one that's still in early days.
It's definitely interesting.
On a more positive note, or a positive note, one thing that struck me with this is how refreshing it is to have a flow of information. I mean, how much we humans enjoy learning. And it's too bad we don't have a MedHow-free university on Zoom and at Cove 2 or wherever we do it, you know, where insights into our planet, our just astonishing planet, are offered, and people come and are interested. I mean, that struck me while we were, you know, just to have this opportunity to have Andy talk to us about this methodology.
Isn't that what the Methow Naturalist is? I thought that's what it was a university. Yes?
Not free.
At $10. So it would help me. You know, I don't know if there's going to be more discussion. If there's, I don't want to stop it, but I have to give this, or I'm giving this talk. This is the first time I've given it. Anyone have any suggestions, things I didn't do as well that I could improve on, or did I go too fast? Any thoughts?
It's very difficult to understand that the complexity, or maybe it's incomprehensible. You know, we act, I think you even said it at one point, we act as if we understand these things. You know, the whole arrangement of DNA and passing on information. But I don't think most of us don't really understand what you're saying.
Yeah, you know, I was going to talk about embryology a little bit. The first time I really got excited, I'm sort of was a science nerd for a long time. But when I took embryology, we dissected the chick embryo and the human embryo, and they're identical to about 6 to 9 weeks. They look the same. You can't tell them. And in the 1800s, when you know, when Darwin came out with his idea, there was a morphologist put all the embryos up of many different animals and showed that they're identical. And his idea is, as they went to eventually creating that, his name is Henkel, the final product, they were going through their evolutionary process that they went through to become that final product. And it was that sort of, I don't know, it made me sort of think we were looking at creationism. But just because we see that DNA, what then programs it to do what it does? I mean, sure, we see the proteins and whatever, but sometimes we build a turtle, sometimes we build a human, you know, a pig, a chicken. It's astounding. We don't create life. We always just take life that's there and then mess with it. There is something bigger than all of us, obviously. We all have an idea of what it is. Anyway,
Well,
any other suggestions?
Yeah.
Was it understandable what I talked about, or?
I thought it was very well done and very understandable. I really enjoyed it. And
Thank you.
Yeah. I wish
Here's a comment. Sorry, go.
I wish we could use this technology for preventing diseases. That would be, and I hope it won't. They won't stop that. I'm sure people who had sickle cell anemia are very happy to be cured.
I treated sickle cell anemia in Chicago a great deal. As you know, it's mainly in Black people. And there is a woman who volunteered to have her genetics altered. And she was a homozygous, meaning she was a very serious sickle cell person. And she's completely cured of it. It's amazing. It cost $1 million to do it in her. So it's not something that can be rolled out at this point. But it does have promise. for that. Huntington's chorea, just a lot of diseases.
Right.
Polycystic kidney disease, and presumably we'll get there. How we'll pay for it is another question,
Yeah, but if we could cure, for instance, Alzheimer, which costs us now millions of dollars.
Yeah, I remember when my mother was my age, she told me, I don't care about AIDS, I care about Alzheimer's.
Yeah.
How about death? How about death? Could you hear that?
I don't think we want to. I think that's a terrible idea.
Yeah, that would not be good.
I do too. I think you're right. I think the fact that
Programmed
we're limited.
obsolescence. Right?
Oh, I just wanted to make sure you saw this one comment in the chat. Excuse me. This is from Vicky to you, Andy. Thank you, Andy, for your introduction. It was really helpful to increase my understanding prior to listening to the TED Talk. So there's some feedback. Yeah, thank you for putting all that time, and Andy really put a lot of time and effort into creating those slides and those, deciding what little snapshots to share. I love your passion just for the beauty of the invisible world, and trying to help us understand a little bit more that all the cascading effects as you talked about.
I mean, proteins are amazing. Now when we look for chemicals for medicines, there's sort of these lock and key systems with proteins where one protein locks with another and then causes a reaction to occur. So now when we're looking at things like osteoporosis, they have the macromolecule of osteoclasts and blasts, which create and destroy bone. And then they create medicines that fit into those. So there's a lot of really interesting work being done where we do sort of understand the system and look for models that will either slow something down or speed something up in reactions in the cells. It's a very interesting science. I'm still a little bit of a science nerd, though. I think I like the Methow better now than the laboratory.
The Methow is the laboratory.
It is. It's the end product. You're right.
And you're in the experiment.
Yeah, it is a very interesting notion of, as you were saying, Tuck and Dana, just, how to bring up somehow some other side of ourselves as humans that can handle these technologies. I mean, clearly splitting humans learning to split the atom really put the whole of life at a huge danger point, and we still are trying to manage that. And Einstein was such a high-minded person, and he was actually a very philosophical person, right? And he had some interesting things to say about how to shift humanity's consciousness into a different. You have to come out of the consciousness that you were in that created the problem.
Well, as you may recall, he famously, or maybe infamously, wrote a convincing letter to Roosevelt, though he was a pacifist, encouraging Roosevelt to initiate the Manhattan Program, and subsequently regretted it for the rest of his life. And I think, there in a nutshell is a sort of the duality of our better angels and our less better angels, which, when I see someone like this lady doing a TED Talk, I think she's looking down the bore of that implicit or intrinsic contradiction in ourselves. Right. I mean, I think we're all thinking you could do evil. This is a great technology. You could do great things for humanity, and you could also do terrible things. And can we manage to do one and not the other? I don't see any evidence in history to suggest the answer is yes.
Yeah, I agree.
Yeah, well put, well put.
It just seems to me that so much comes back to our relationship with power and the necessity of humans to reorient themselves and make sure that we don't have people in power who are actually psychopaths. I think you can do a test for that, can't you?
That's an oxymoron.
That's right. That's true. There's a really wonderful video, The Fog of War. I don't know if anyone's seen it, by Robert McNamara. He talking about our involvement in World War II, bombing Japan, and then later in Vietnam. It's, you know, a real admission from someone who was in charge of all of that, of what really went down. Very scary, but very honest as he was getting older and dying, talking about US's role in some of the things they did in those different countries.
Yeah. Any other thoughts out there? Last few minutes.
One more thing, and I just wonder if this touches the scientifically oriented people in our little group here. There's this term that the discipline of big history, which is the evolutionary story of the universe as it's taught in schools these days. They call it big history. They use the term, rightly so, that it's an emergent universe. So the word is emergent. It's an emergent universe. Like I think Tuck said at one point to Andy, you're in the experiment. The experiment is not done. It'll never be done. It's an ongoing process. Anyhow, everything that exists has emerged, and everything that humanity is capable of has. Humanity didn't exist. Now it does. It has these capacities. We're not done. And so, Tracy, this is something I know you can relate to, and I don't know about others. The potential for humanity to live well on the planet is staggering to me. The potential, because we're capable of loving life. We're capable of it. It's elusive for us, for maybe all of us, but the potential of emergence, of gaining higher ground, I think it really does. To me, it actually exists. It actually gives me goosebumps. I don't see it happening. You know, what I see is the war in the Ukraine, more like, and people starving to death, but the potential is there. Everything else that exists has emerged. There was nothing.
Yeah. And I think too that there are some incredible things emerging. There is some, you know, there is incredible wisdom within humanity, incredible, you know, that does exist. It doesn't necessarily garner the headlines the same way, but because there is just some really horrible sides that also we're coexisting with that definitely take our attention necessarily, as we should be paying attention.
There's an interesting book that I just finished called Factfulness, which basically is written by
Andy, for some reason we can't hear you. Tuck, were you going to say something?
I could hear Andy.
I could hear Andy.
There's a book called Factfulness, which is quite optimistic about what's going on, and just talks about how things are really better than they've ever been in many ways. Not that there isn't a big need for a change, but it's written by a public health physician from Sweden who just recently died. It's his kids who published it. But it just talks about the fact that there are not a first world and second world. There's 3 different worlds, and how much most societies are really advancing and getting better. It's more optimistic if you want to have a pretty factfulness look at the world that isn't so negative. Anyway, I enjoyed it.
What is the name of the book? And
It's called Factfulness, F-A-C-T, fullness. And it's, I think the first name, it's Hans something, the author. He was a public health physician, worked in Africa, many different places from Sweden, and basically puts humanity into 4 categories. 1 billion of us who are at the top, billion of us who are at the bottom making less than $2 a day, and then up to, I think, $16 a day, $32 a day, and then talking about the different ways their lives are and how life is getting better for most of us, and that the population of humans is probably going to only go to about 12 or 13 billion before it stops growing, which is way too many, but is less than I thought. I thought it was always an exponential rise forever, you know, until we sterilize the planet. But that doesn't seem to be the case.
Yeah.
Well, there could be a lot of interesting conversations springing out of this one. We are at 10 o'clock, and thanks so much, Andy, for bringing this to us. And I think, yeah, it's just been great to see the reception, and thanks everybody for joining us here today. Any last words before we sign out?
Thank you, Andy.
Yeah,
Thank you, Andy.
yeah, thanks for coming. Great to see you all.
Yeah, thank you very much.
All right. Have a great day, everybody. Enjoy the magic of life. It's beautiful.